Okay, I'd like to welcome everyone back to the last session of our E340 542 class. Today, you do all the work. I just listen. I would appreciate people listening and commenting, asking questions. I know people work together pretty hard over the weekend, and I want to thank Elmer for suggesting how you could publish these. I think that's a great idea, and I hope everyone will go and get a DOI for their project. As always, this material is live streamed and recorded. If you don't want your section of the presentations presented on live stream, then let me know and I'll edit it out afterwards. I thought that since Elber seems to go last most of the time, we would start with Elber today, and then Ibrahim, Pedro, and William. So I tried to randomize the order a little bit for the presentations. And so today we'll start with Elmer, and I'll turn the screen share over to you.
Mm-hmm. Okay. Okay. Can you see my this? Do you just see the Grand Canyon, or do you see my screen?
Yeah.
What do you see? Do you see my
GitHub homepage?
GitHub repo. Okay, great.
Yeah.
Great. Yeah. Okay. So I will start with my GitHub homepage because that's where I have the whole project, and then start the Colab notebook. Go through the notebook. I realized when I had my feedback with Williams that I have to really explain what I do in the introduction, because it's not directly what people here are familiar with, even if it's very, very close to what we did. And I think first really have to be clear what I do, that you understand the results that I produced, and we'll see that we really a lot of ground already covered in class, and this is just a little bit a different point of view and an add-on. So I start here with my GitHub account. So what I did is that I have a README page where I as well generated this DOI, and here I have as well even citation, and the notebook is of course here, and you can start it. It loads, but then you can press this open in Colab button, and I did this already, and then it opens in a separate tab. I already run the first cell that we have to do to install the C libraries, and I already run cell where I generate my controls to control the widgets, and I load my Python libraries. So let's go to the introduction. So this is my project work from our class, and as you all know, we have used Tellurium, and we have studied biological networks, and I want to first give some biological background from the network that I studied here. It's a Boolean gene regulatory network, and it's about mulate blood cell differentiation. And I have here a very detailed plot about how hematopoietic stem cells, blood cells, divide into their different cell types. And there are two major branches. One is the lymphoid branches, that it's in our lymph system, and and all of the cells that it differentiates into belong to our immune system. And the network that we study and my presentation is not focusing on these immune cells. But then there is as well the myeloid part, which is ripening in the bone marrow. These cells are ripening in the bone marrow, and they are as well differencing in different blood cells. So we have as well in this part of the branch immune cells, and these are the macrophages, which are monocytes that invade, so monocytes. We have the granulocytes. These are as well immune cells, but then we have as well the erythrocytes, which are the cells that transport our oxygens from the lung into everywhere in our tissue. And then we have the megakaryocytes, which will turn into platelets, and platelets are used that when we cut us, that the wound heals again together. And what I highlighted here with these blue boxes are the major, what will be a attractor in our system, the steady state point that our cell differentiation can reach. And they are as well highlighted here. This is from the paper. So we have this common myeloid progenitor cell type, and this can differentiate either into the megakaryocyte or erythrocyte direction, and then into the cells. This is this branch, so it's just mirrored here. And then it can as well go into the granulocyte, monocyte direction and branch like it is here. So this is what we will now have a look at. So far, the biological background. Take quickly step back to the mathematical background. So we have Boolean. We study here Boolean chain regulatory network, and the functions we use to simulate to make step functions with ordinary differential equation. We use Hill functions, and the Hill function, it's here the output is Q, is a baseline noise of our product, which is Q, and then we have here this formula that has the maximal the expression of gene A, but it would actually be the substrate. That's the A. Then we have a baseline expression rate of the gene Q. Then we have the Hill power, and with the Hill power we can specify the steepness of our curve. We have Vmax, which is the maximum reaction rate, so the velocity when the gene is on, that we can specify. We usually take 1 in our case. And then we have the Km, which is, if we have a Hill power of 1, then this is actually the Michaelis-Menten constant. But because we use as well different Hill powers, in my study I used Hill power 3, because that's the one that I used in the paper. It's not directly the Michaelis-Menten constant, but the idea of this Km is that the gene is switched on or switched off when it goes over the threshold that we specify with Km. Usually we specify 0.5, but in the paper they sometimes are used as well 0.3, and actually when you do the translation that we did with the enzymes and we came to the Hill function, there the threshold is as well by 0.3, I realized. Anyhow, I have used in this study 0.5. Then we have Kd, which is the decay rate. So the protein decay that we have, which is a first order kinetics. And as I said before, we have the baseline rate we set to 0, Biomax we set to 1, and through that the Hill power function reduces to this form. This here shows a plot from Hill function, and we have different Hill powers, and you can see first order is like, it looks like Michaelis-Menten, and then you have this step function that gets more and more extreme as higher the Hill power. Okay. So, and now I come to the part that I think the people here are not that familiar, and these are the Boolean gates. And as discussed before, we looked at the AND gate, and the AND gate means if gene A is on and gene B is on, then the gene Q will be on. But if one of them is off, then it will as well be off. And we can, with the Hill function, with the Hill equation, we can do that by multiplication. So it's a product that we take from both of the Hill functions, and through that we get the resulting value for our gene that we steer, that we control. And these hex plots, they are showing exactly the same as these truth tables. So we see here the gene A, and we see when the gene expression is high from gene A, and the gene expression is high from gene B, then the expression from our gene Q is yellow, is as well high. But when it's low, or when one of these genes is low, then it's more in the dark side, and that means it's more towards zero. They show up a little bit too big here. They are smaller local. So this is how in electronics, when you lay out a switchboard, the symbol, how you represent an AND gate. Then the other gate that is often used in electronics, but as well in our in this translation to the Boolean network, is the OR function. An OR function is always on when one of the gene is on, and it can as well be both of the genes on. And as you can see, that is exactly what this hex plots as well is representing. And this is an OR function we build with the sum of with the sum. So we have to add the two Hill function together. And there would be a different way to do it. And what I have chosen here is the cooperative non-competitive binding, and that's similar binding like we have in myoglobin, where we have when we only can bind one oxygen atom, but there would as well be the competitive binding, like we, for example, have in our blood cells, where we can bind more than one oxygen molecule. But I have chosen this one, especially when you plot the other one, you will see that this hex plot, that you don't get this nice square that we get, that is kind of the opposite from the AND square. And I want to translate the model, I thought it's better to have really similar kind of gates. So this is the ANSI symbol for the OR gate that is used in electronics. Further gate that we will use for translation is a NOT gate, and the NOT gate is just an inversion. And because our Vmax is 1, we can just subtract from whatever we have, and then we will get the invert, which is the NOT. So when we have something that is 1 and we subtract, we have 1 minus 1, then we have 0. If we have something that is 0 and we have 1 minus 0, then we have 1. So that's how we can build the invert. In class, we have done it that we flipped we took Km to the top. So that was another trick to do it, but I prefer the way to 1 minus. It's just what I'm more familiar with. And then there's the proposition gate. This is actually not a real gate. It's just Q equals A. So it's in one case used when one gene is just really following the other gene, and when we kind of have a full correlation. So now we come to the gate that is most commonly in biology used, and this is the no implication gate. And it can be as well written as B and not A. And that's exactly what you can see here in this hex plot. So when B is high and A is not high, then it's on. And all the other time, it's just off. So this is the truth table. And as you can see here with the Hill function, we set the whole things exactly together. It's like an AND. So we have a multiplication, but then we have as well not A. So actually this should be here B and this should be A. I just realized, so, but it would be A from the Michaelis-Menten function, then 1 minus A here. So this should be flipped, but that's not what we have. And the gate looks like this. So always in a circle, it's a not. So not A, but B, and in the end, we have the invert because it's a non-implication gate. If we not would invert in the end, then it's just an implication gate. Okay, so this is the background and I come from electronics, so that's why it's something that I'm very familiar with. And if I can map my biology to electronics, then it gives me a very nice tool to understand what goes on in the network. Now, Stuart Kauffman have in the 1969, he published his first paper where we used Boolean gene regulatory networks. And he specified here, just as an example, 3 genes, and he specified exactly these Boolean gates that we discussed before. So we have here a non-implication gate, we have here an implication gate, it's just opposite, and we have an AND gate. And we have here 3 genes, X, Y, and Z. And when you have defined for each gene the Boolean gates that drive this gene, then you can from this one extract the network topology. So first you can extract the transition table, and as you can see, this is derived from when I have 0x and 0y here, and then my C will be 1. And when I have 0x and 0c, so x and c is here, then I have 0j. And when I have 0j and 0z, then I have 0x. And like this, I can deduct the whole topology of the network. So first transition table, and then I can from this one come to the kinetograph, which shows us nicely the dynamics. So from each state that we have, from each transition states towards how we come to the attractor. This is here a little cycle attractor that we end up in this case, in this example. This is quite okay when you have a handful of genes, but of course you will run into an exponential problem when you have 2 to the power of 100 genes, then you cannot anymore do it. There are different other techniques used to find the attractors, but for our example, this is still good enough. So we'll end up with about 256 lines of, you will end up, it's about on the limit to do it like this. So when I looked into this project, what really took me very, very long is I know how Kauffman defines the network, I know electronics, but really to bridge this way, how he derives the network topology and the transition table from how he sets together his gates, to really understand that how it's done in biology, that took me quite a while actually. And the easiest to see in electronics is called a flip-flop, and this one is done, it's a set reset flip-flop, and it's done with not OR gates or NOR gates. You could do the same with NAND gates, not AND gates, they are. And this here is, as we the transition table for just the gate. As we see it's just an OR that is inverted. And we have here a kind of a set input, and we have an inversion, and then we have another inversion here. We would have, if we want, a reset input, and this is fed back to our NOR, to the other input of the NOR gate. And like this, we have in this flip-flop a memory state, and we can toggle switch between 2 states. And what the big click was, what I needed to see how I can translate this, was that you really have to have this set and reset state, and then you have the gene input, so A and B. So this is A, this is B is my output, and then you have here in the transition the current state, and this is the next time step. Now I have quickly to check what time it is. Yeah, I still have some minutes. Okay, so you take these set and reset states and you copy them just to the output. We don't have to do anything with them, but then you really take the states that A and B and what will result as we did before to our A and B. But what we look at is only at our output B. We not much care about the A output. And we will then end up in a memory attractor. We have here this reset B to 0 attractor or set B to 1 attractor. That it doesn't matter if we click the S, or if we set S, or if we set R, it will always either reset B or set B to 1. And then if we press both at the same time, then we have still a reset B to 0, but you will see what happens. So this is just a transition table. Now we can as well do the kinetogram out of it, and we will see that we end up here as well in such unstable attractor states. But as soon as we then click again one of the S or the R, we will go back to a state that is known. And like this, we can control the whole toggle switch and have the reset to be attractor, the set to be attractor. We have the memory attractors that we need. And so when this was clear to me, then I could actually go back to what we did in school here in our class. And there we had this toggle switch, you remember maybe. And this toggle switch can be implemented molecular genetically, that you have a promoter, you have a receptor, and then the receptor will block promoter 1, the promoter, and then we have the receptor 2, which will block the promoter from promoter 2. And then we have these inducers. Our latch, or what we call latch there, or our flip-flop looked like this. But actually, what I really had to see is that we have as well this set and this reset, and we not explicitly put this into these diagrams. And these are small things, but sometimes you exactly need them that it makes click. And here they show the inducers. They show them the S and the R. But anyhow, so I translated this one. I calculated the gates, or made the gates, and of course the Boolean function, no surprise, is a non-implication that we get. So there are two non-implication gates that this gene A and B are to each other. So you could as well make a flip-flop, but this time not with NAND and not with NOR gates, but with NIMPLY gates. And you can again draw the transition graph, and you can study your kinetogram, and you will find the same number of attraction states, but they're at a different place a little bit. So you have a memory attractor, you have an unstable attractor, you have a set and the reset attractor, and you have here the other part of the unstable attractor. It's a little bit different, split it up. But this is really how you can bridge from electronics to biology. So, and that's exactly when I did this course. That's the point where that's what I wanted to understand, the major thing, of course, other things too. So, okay, and I come now to the model that I really looked at. So this was only background information. So this was the model that was in the paper, and we have 11 genes. And as you can see, they specify each of the genes, and they always specify the Boolean gates that steers this gene. There are, let's first focus on this FOG1, GATA1. This is the case where really FOG1 is just doing exactly the same as GATA1. So this was the case that was just sorry, I forgot the name. The simplest case. Anyhow, not yet. The proposition. So we have just A is Q, and that is exactly what this case is. So we have the proposition case here, and then this EKLF. This is an imply gate. So you have GATA1 minus 1, GATA1 and not free 1. GATA1 and not ELK-F. It's as well a non-implication gate. The SCL is as well a non-implication gate. All of these are non-implication gates. Then when we look at this CEBP alpha, we have again a non-implication gate. So we have CBP alpha and not, and then this is a whole other expression that is under the hood, but in the end, it's just an and. So it's got the 1 and FOG1 and SCL, and we take the invert of that. And so on the big top view, this is a non-implication gate as well. Same here, non-implication gate, but a little bit special because we have here an autocorrelation. This Pu.1 is as well a gene that auto steers itself. And we have here OR. This is an OR and this is an OR. So they are not ANDs like they are here, they're ORs. And same here. So non-implication gate with an AND, non-implication gate with the AND. Then we come up here and there are a little bit more complicated gates. So we have a non-implication gate, but then as well AND, not PY. And here we have a non-implication gate, but we have 3 ORs and they are as well autocorrelated. Now, this is all based on literature, and they have even in the supplementary data more information about how they derive these gates. And this is when you now do the graph, when you now generate from this one the graph, like Kaufman did, this is the graph you end up with. And interestingly, the graph that they describe here, and it's as well the graph that is implemented in the software that they analyze, looks like this. Actually, the graph that they put into the paper looked a little bit different in this corner. But anyhow, I went with this graph that really is what was implemented and what the genes specified. And I made here some notes. So, for example, FOG1 and GATA1 is AND connected with GATA2. So AND connections are OR connections. So everything that I saw here with AND and OR, I highlighted here with these little tags. So also in Stuart Kaufman's paper, when we have the graph, we can make the state space or that we have the dynamic graph, and this looks here like this one. So it's not anymore that we have, going back here, it's not anymore that we have it directly like this. It just shows the big picture, the big tree that we have. But we went exactly to the same state. We had first each gene defined the gates. We came from that to topology. We don't really do the transition stable, but we come to a state space that we now can analyze. Okay, good. And this is as well from the publication. So this is the output that comes from the gene regulatory network. So the idea is you have different inputs, and you will end up in one of the 4 major attractors. So either they are erythrocytes, megakaryocytes, monocytes, or granulocytes. So each of them is one of the attractor. Each cell type is one of the attractor. Each differentiated cell type. And this is how the gene should be expressed. So this is how the implementation from the whole model looks like. And I think you know in the meanwhile Tellurium too. So let's run it. And of course it gives some error. Not, oh, come on, please not. Let's see. What is going on? Ah, come on. I don't know. Okay, great. So it doesn't show my vigets, which is bad. Oh, come on. Okay. Give me 1 second, please. I think I know where the problem is because I developed this locally, and this is now a little incompatibility, which is stupid. I don't know if I can fix this. This is now bad. Okay, well, this is okay. Give me 1 second. I'm sorry. Um, with this one. Okay. Okay. So, okay. So this is how it should look like, and it's because of there are 2 different versions of widgets, so that's why it didn't show up. So more or less, we have here 2 tabs. One is all set or non-set, and what we have now to try to do is we do the right setting here. And in the beginning, for the first run, let all the cells set. They would be all on, and then we run the network, and you can see our input was all on. This is our time series, and this is how we end up with which genes, how they are expressed. So SPI1 will be expressed, CEBP alpha will be expressed. Oh, my battery. Okay, I hope the battery charges. Yeah, yeah. Okay, and then, gosh. Okay, so these are the genes are expressed in the end. So, and when you now go up, I have to find the mouse again. So when you go up, and when you now compare this to this one, you will see that actually, if you do no settings, you end up with monocytes. When all the genes are expressed and you just run the model, then you have here CBP alpha, PUI1, June, and ER, EGR, NAB. That is a whole complex. So, and this is what we end up here, if I remember right. So the exercise that we have is that we have now to orient ourselves on this state transition graph. And if we take the right settings, so if we take that one on, ZFM one on, SPI one on, TAL one on, and then we choose one of those one, ELK F1 on, or FLI one on, then we end up in one of these states. And we can do the same that we do them all off. And we, for example, do GFI1 on, and then we would end up with the gene expression that is the granulocytes. So, okay. Because this is now not going how I planned, I think I can do one example. Let's see the granulocyte. So GFI should be positive, but I think only GFI will not be enough. So I think we have as well to cut out 2, for example, to put it on. I don't know if this is now enough, but let's see. Let's run it. And oh, yeah. And you see, we end up with GATA1, ZMFP1, and TAL1. And when we then go up here and compare this, GFI1 should be up. I have to, PUI1 should be up, and E or B2. No. Okay, this is now not good. So, okay. Okay. I'm sorry. Okay. Anyhow, the exercise would be that you take the state graph, you investigate it with what you know from the state space, and you should end up with the gene expression, even if this at the moment doesn't work. And I think the implementation is right. I got to the right results. I can at the moment not reproduce them. Okay. But I want to show as well what I have otherwise in the notebook. And I can at the moment not explain myself why this is not working as it should. Sorry. Okay, let's go to the gates. So I looked at each of the gates. So this is the proposition gate. And I did from each of these gates. When you run it, you get the time series. I take the first of these numpy gates. So there you would get the information from how a numpy gate looks, as we have seen before. And then I come to these are the numpy gates that actually had autocorrelations. And these were kind of interesting because, of course, they have a for these gates, I have as well done the phase plots. And you will see that if you only look at the output, so when you have GOTA1 and FLI1 as an input and SPY1 as an input, then there is a part of the output that we map to that is not not touched at all. So we have there a small phase transition. When we look at the input, when we not take the steady state for the inputs, but we only take we take what we do as an input and we look at what we get as steady state as an output, then we get the whole picture, of course. And I did this because when you measure gene expression experiments, then you have usually a snapshot. And in this case, you only have the steady state values. You will never have the input value and the next value. So you only can measure a snapshot. So the only thing you can measure is really that, even if this is from one to the next step, what we get in the Boolean case. Okay, so there are other gates that had autocorrelation and were all NumPy gates, but then there was as well one NOR gate that we had with 3 inputs, and this one had as well autocorrelation, actually quite extreme autocorrelation, and this was more or less like giving a very, very narrow on state and the strong on state, and the rest was all off. That was what it regulated. And after that, I looked as well into the motifs. So we have here the GOTA1, GOTA2 fog motif. This is one cycle that we had. And I looked into this. And I can do this in them that I set, for example, GOTA1 on, and then I can study what happens to all of the genes, or I can only set GOTA2 on and all the other ones are off, or the fog1 on. And so the first one is just that GOTA2 is on. Why it takes please. Yeah, it should plot what I have, but at the moment something is messed up, which not was the case in the afternoon. Let me set everything.
Okay, you should probably try to wrap up.
Yeah, okay. I'm sorry that this is not working. This is really not what how it was when I test on. So Okay. Anyhow, I studied each of these states. Let me quickly see if this one runs. Yes. So example, again, here you have the input, you have the time series. This is what you get, what the output is. You can set another input, and like this, you can really, here all of the genes are off. Like this, you can explore each of these modules that we have. And then there is the third module, same way, and the last module the same way. Yes. So I could do quite some exploration. I'm sorry that this didn't work out this time. These are my references, all the papers that are somehow were used for the study. And that was my presentation. Yeah, thank you. And if you have questions, I'm happy to answer. And I hope that you have seen the bridge from what we did at school and the bridge to electronics. Yeah.
Okay, thank you. Some
Yeah.
And I put a few questions in the chat from me.
Yeah.
I think one thing that does maybe not so clear is what is it you're trying to explain with this model?
So the model itself does explain the mirrored cell differentiation, right? And I try to understand how the electronic switches are related or can be seen as these Boolean switches and are related to biological switches. I try to bridge this, and I try to explain this in this notebook.
Well, I guess
but you ask about the model.
So when you say explain differentiation, that could mean a lot of things. You mean the particular sequence of differentiation states you see? What is the how would I use?
Yeah. So a differentiated state is a cell type, right? So when you differentiate in different cell types, and each of these cell type is an attractor. And it depends on your input setting. That's what, I'm sorry, it didn't work in the demo. When you set your input state, then it will go to a sudden attractor. And what you can show, and what I could do when it was working, was that I go to each of these 4 attractors, so I can reach all of these 4 cell types, and that's what this model tries to show.
So starting from any initial state, you always get one of these 4.
Yes.
And I guess, again, I'm trying to understand how you maybe use the model. Would you be studying perturbations? You talked about knock-in or knockout, maybe.
It's only expression, and we don't have to. I mean, when cell differentiation happens, it doesn't have to be a knockout gene or not. It just depending. It will just depend on what values you set that we can do actually in a model, and that was what is at the moment not working on Colab. So when I set the values here, in this case, so according to what values are set here, I will end up in one of the cell states. How I can steer them by clicking them on and off. Of course, in the living organism, and it would be different. It would be hormones that steer that, or other genes, or we look here only at the small part from the whole network, of course.
Well, I mean, I'm trying to understand what you could do with this model. You've shown very nicely how you build it. But I'm trying to understand what we learned from it. So one thing you might try to learn from it is how robust it is. If you have some external perturbation, do you come back to the same state or not? You could try to understand how noise resistant it is. You could understand how fast you differentiate, how stable differentiation is.
Yeah.
Many
yeah.
questions one might ask. And so I've tried to understand what kinds of questions you're interested in and what you want the person who does this exercise to try to understand.
I try to show to the person. I mean, it's not clear when it's a little bit more complicated than just a bistable switch, right? You have more places where you can end up. We have 4 places in this case. And I tried to teach him what are the input settings that he can choose to end up in one of those states, and that he always will end up in one of the states. Of course, it's simple, but it's more or less that what the child regulatory network does. I mean, I could have had studied the stability or something like this, but I didn't went that far. For me, that's not as far as I came. I was happy to set up the whole model and be able to look at all the phase plots and all the bifurcation plots from each of these elements. That was what I more or less did. Yeah.
Okay. Any other comments or questions? I mean, there's a lot to discuss here, but I think we need to get through everybody today.
Yeah.
Does anybody have, I mean, an urgent question we should certainly allow. Does anybody have another question? Ibrahim?
Yes, look like.
I just was a little confused what the point of a NumPy gate even is. If it kind of ignores one of the inputs, what's the point of even having it as an input?
So, I mean, it's the switch that is very easy to do in biology, and through that it exists as well a lot in biology. So it's molecular biologically easy to do, but it's as well easy to do as it's that what is most occurring in biology, and that's how biology regulates itself.
Is it also not just, I guess, just like the promotion of one and just the ignoring of the other?
Well, it's not totally ignoring the other, right? It's and not. So it's specific. I mean, okay.
Because in the graph, it shows the one that you have. It shows like it is high when B is high only. And if A, regardless of what A is, it doesn't matter. And if B is low, it's also low. So can you not just simulate that by saying that if B is high, then the output is high and just ignore A altogether?
No, not because there is as well, it's like an AND gate, right? So where we have it. So it's as well that it's only if the one is low and the other one is high, then it's high. It's not so that it's exactly always like the other one is. So when it would be like, it's an inverse, right? It's you see, if it, for example, just would be like B, then it would be high here and low here, but here B is as well high and it's low, right? So it's like an AND gate. So you have always one condition more. Yeah. So and it's not so easy from the protein and from how biology works to make an AND gate or to make an OR gate, and not the nature have just chosen to make NumPy gates, right? But it's what we find. It's just because evolution have generated them. And in electronics, when we did our engineering, it was just much more easier having tubes and later on semiconductors to make AND gates, or NOR gates, or NAND gates, and do all the circuits that we have with NOR and NAND gates. And that's what is standard in industry. But biology had other things was easier, and evolution have chosen other things, right?
Okay, got it. Thank you.
Yeah. Okay. Yeah.
Any other comments or questions? Thank you, Ibrahim.
So my name is Pedro. My project is about Nipah virus antagonist interactions with innate immunity. So this is a replication attempt. The word attempt I will explain later, but the objective is to replicate the model they used in their paper to model, describe the results they got experimentally. First, an overview. My references, there's the main paper from Seto, and Tellurium, of course. I'm referencing the documentation, and the link to my Colab notebook together with the DOI. And here I generated the DOI as Elmer suggested. So thank you, Elmer. It worked and it was simple. So let's start with the biological background. What is innate immunity? I think this is the core concept here, and this is the objective, the learning objective of my project. First of all, cells can detect viral particles in the cytoplasm. They can detect other particles from other types of living or non-living beings out there, but focusing on viruses like Nipah virus, influenza, etc. These viruses, they release proteins and RNAs inside the cell that are different from the host. So cells have ways to detect those new RNAs, those alien molecules, and activate a defense system. So first of all, by detecting those particles, they activate the production of cytokines, for example, TNF-alpha and interferon alpha and beta. These signals are sensed in an autocrine way, which means they sense what they produce, and they also sense what neighboring cells produce. This autocrine sensing amplifies the effect of they amplify the production of cytokines. So it's exponential effect, exponential production of interferons. That's what leads to inflammation. When they are exposed to such high levels of cytokines, cells start then to produce peptides capable of destroying the virus and inhibiting viral replication. So this is very important to contain a viral infection, mainly in the first stages of the infection. Later on, generally, the adaptive immune system take over, but in the first place, and also to alert the adaptive immune system, you need the innate immunity. Nipah virus specifically, it has 3 proteins, V, P, and W, that suppresses innate immunity. This is one of the reasons why some researchers think Nipah virus is so deadly, because it can infect you and replicate inside tissues without alerting the innate immunity. So in their paper, they studied, they developed a dummy virus. They've taken NDV, which is Newcastle disease virus, which doesn't have, or I mean, they took off the V genome encoding. I phrased incorrectly, but who cares? So NDV without the V protein, and then they added either V or P or W genetically in this virus, this dummy NDV virus. So they could study the effect of P, V, and W separately on the innate immunity response of dendritic cell cultures. What they found is that P, V, and W proteins have the ability to suppress innate immunity in the following order. W is the strongest one, V is the mild one, and P is the weakest one. So as you can see in the levels of interferon beta, interferon alpha, and TNF alpha, these are all cytokines. You can see how, so the red is the dummy virus without VP or W. And as you add each of the proteins, either P or V or W, you get diminished responses. The pathway that describes what's happening comprises of, first of all, the detection of the virus by, so the detection of the virus, I will explain how it's modeled later, but the detection of the virus triggers production of TNF alpha and interferon beta. TNF alpha has a positive feedback loop. So you release TNF alpha, you sense TNF alpha, it regulates the production of more TNF alpha, so on and so forth. P, V, and W proteins, they act on this circuit to suppress the production of new TNF. So I drawn a small, like a summary of this this pathway here in a very simple way, and this is more faithful to the equations rather than to the diagram. So it's basically two species, TNF alpha M and TNF alpha in the environment, and one activated the other, while P, V, and W suppresses the activation of TNF alpha mRNA by TNF alpha in the environment. The other interesting thing here is the positive feedback loop on STAT circuit. The STAT circuit also is a positive feedback loop. STAT activates STAT P2, that activates STAT mRNA, activates STAT back. So this also, this one here, so, okay, I have to come back a little bit. So in the TNF alpha, we would expect this to be a binary system, bistable system. It's not in their implementation because TNF alpha in the environment does not decay. On the other hand, the STAT circuit, it has decays and Michaelis-Menten equations for all of them. So this here you can achieve low and high levels of STAT. And these two networks, these two circuits, TNF and STAT, are coupled. So there are three ways they are coupled. First of all, by IRF7, which is interferon regulatory factor 7. When you have high levels of STAT, you trigger the production of IRF7. That triggers the production of TNF alpha. That triggers the production of STAT. So the purple arrow symbolizes the TNF activation of STAT. And then the blue arrow is an activation of IRF7 on itself. The other way it is coupled is by interferon alpha and interferon beta. IRF7 activates production of interferon alpha and interferon beta that by themselves activate STAT by autocrine signaling. So there are many equations. There are 14 equations. I decided to explain 2 of them because the others are very similar. So the first one is the production of interferon beta, which is basically a Michaelis-Menten equation. So the rate of interferon beta in the environment, so it's also considering secretion, both production and secretion, is a Michaelis-Menten function of interferon beta mRNA. There is no decay here, which means interferon beta in the environment is produced and keeps growing to infinity while you have any level of interferon beta. If interferon beta is 0, you keep interferon beta in the environment in a constant rate, in a constant value, sorry. The second equation is similar, so we still have a production rate in the Michaelis-Menten form, but we also have a basal production, which is R1, and a decay. Minus TNF alpha M means that TNF alpha M decays. As you can see, R1 and the Michaelis-Menten function are multiplied by a coefficient IC. This IC basically multiplies all productions in the equations. So almost all of them. There are a few exceptions. So basically what's going on is that IC models the interaction of the P, V, and W proteins in a way that I will show you later. So it's a scaling factor. If you have lots of P, V, and W, what is expected is that you decrease production of TNF. The presence of the virus is modeled as a linear increase in proteins P, V, and W. So they do not model viral cycle. They do not model virus unpacking, replication, anything. The presence of the virus for them means that P, V, and W proteins are being produced in a constant, not in a constant. Yeah, they are produced in a constant rate, which means their levels increase linearly. So P, V, and W are equal to a constant, R5, times T. T is time. You can think of them as our substrate or as our non-homogeneous part, the non-homogeneous part of the system. IC, which is that factor, that coefficient multiplying the TNF alpha mRNA production, that factor depends on P, V, and W levels. P and V proteins, they are inhibitors, and they behave in the following way. So take this first term here. 1 plus a constant times P divided by 1 plus P. This function goes from 1 to SP1, this coefficient here, this constant. And this constant is lower than 1, which means that as P grows, as P increases, IC decreases until SP1. The same so the graph I'm showing here in the left. The same happens to V. SV1 is also lower than 1. So as V increases, IC is lower than 1 until it reaches SV1. That's the idea. So they diminishes the production of certain cytokines and expression of certain cytokines, but they do not shut down. They do not shut them down. They just diminish. While W, on the other hand, you see there is no numerator depending on W as the other have. It's just 1 divided by 1 plus W. So as W increases, IC goes to 0. So W has the ability to shut down the cell innate immunity. One interesting detail here is that you can have either P or V to suppress innate immunity. So this multiplication here works as a OR, despite the fact it's a multiplication because of the functional form they adopted. And they also have a stronger effect when combined, because when you multiply two values lower than 1, you get a lower value. So they work as an OR, but their effect combined is also stronger. Some key results. The first result is that as you so you start with the NDV virus, which has a strong secretion of cytokines, and you add each of the proteins, V or P or W. You see that the release of these cytokines, the production and release of these cytokines is much lower. It's not clear from this graph, but NDV with the P protein, which is the green curve, is not so low as the other two. And as we'll see later, V and W are the strongest. Here you can see more clearly in interferon beta and the mRNAs, the order. So NDV without any of the proteins are the one that causes most reaction in the cell. P is the next one, is the mildest of the three. V is the one in the middle, and W is the most powerful. So you can see that W indeed has the ability to shut down innate immunity. Here I put side by side my results and their results. So this is for NDV with W protein, comparing to NIV, Nipah virus, the natural Nipah virus, and how they affect mRNA of TNF. And in their results, they say that the dashed curve, the broken curve, is NDV with W protein, while the continuous curve is NIV, the Nipah virus, the natural Nipah virus. And I found the other way around. For me, is more intuitive for me that combining all three will have a stronger effect. So I don't know what's going on. Maybe they have some mistake, they switched, I don't know, or I did something wrong. And it's very possible because although I checked the parameters and the equations like at least 3 times, it's not peer-reviewed, so it always could be an error from my part. Yeah, they could just have switched the broken curve and the other one. So, yeah. Issues. There were many issues. First one, it was challenging because it were 14 ODEs and 49 parameters. So that's not something you can understand in 30 minutes, and that's not something you can transcribe in 30 minutes. I took a few days to transcribe everything, check the parameters, and even being very careful, I found many mistakes, and meeting with James, we found many more, and so on and so forth. So it was challenging in this aspect. The parameters they provided parameters in mixed units. Of course, it's my mistake to not having detected this issue in the first place, but the the curious thing is that with the original parameters without converting, the curve shapes were more similar to the papers than converting the units, converting all the units to the same units. So this was very suspicious for me, but I went with the converted units. Of course, it doesn't make sense to put mixed units on the simulation. And also, I put this disclaimer on my Colab notebook. The curves do not perfectly match, and the values do not perfectly match. If I think they used the wrong units in the simulations, I think I don't eliminate this possibility completely. I still have a stronger feeling that this was a mistake from my part. But I still leave this option open because I reviewed this like 3 times, and until the point I couldn't find like any more mistakes and any more typos. So I'm still not confident because it was not peer reviewed. We always reach a point where, although we check over and over, we never spot the mistakes there are. The mistakes are there. So that's why ideally you need someone else to review, and I didn't have it, like someone to really go equation by equation checking all parameters, which would be painful. 49 parameters, 14 equations. But I went forward nonetheless. And something that really worries me is that some behaviors do not match what I expected qualitatively. So for example, when I increase a certain parameter, for example, a certain curve doesn't change much as I expected, or if I decrease something to 0, it doesn't change something, which is worrying in my opinion, because but it could be the case that I simply did not fully understand the network. So here, James' advice, which I agree on, is to break down the network into smaller pieces and analyze them separately. But I did not have the time to do this thorough analysis of the network piece by piece and then merging them together one by one. So I did not do it, but I think this would be the way to really solve all problems and maybe detect if they did a mistake or if I did a mistake. Conclusions. Well, it's not a peer-reviewed project, but it's a replication attempt of their model. Okay. There's a notebook, Colab notebook, with widgets that help users to change parameters and see the results live, and exercise that can help the user to understand the model. And as a bonus, I added an exercise that discusses a potential drug to treat Nipah virus. So in this exercise considers targeting the W protein. It's a small discussion, but I think it's worth having. I will show you the notebook. This one. I don't, in GitHub it shows as a zip zip file that I have to download, extract, and then double click the Colab link, so it's still an annoyance. So I'll open the original. I will go through things I changed relative to the last time I presented the Colab notebook, just to be quick. Okay, I won't go through it all. So first one, there's a disclaimer that I'm not sure it really replicates the results. Maybe there are some problems. At least in the explanation, I've broken down a few pieces of this network. So I first explained the TNF-alpha self-activation with PV&W inhibiting production of TNF-alpha mRNA. Same thing for STAT and how they are connected. In the mathematical model, I grabbed 3-4 equations to explain them carefully. So for example, let's look at equation 29. You should read it as na. So I explain each term of this equation and say, so as you can see, other equations are similarly written. So they follow more or less the same idea. And then equation 30, there are interesting interactions between activators and inhibitors. So I explained that carefully, although the real derivation of this function is more complex. So I point to the supplemental materials where they derive this equation here algebraically. Okay. Equation 43 is the definition of IC, the inhibition coefficient by PV&W. I go through it carefully. Equation 44 is the way they model viral presence in the cell. And here in the, let me just install the packages first. What I did differently here was to add options for each of the viral replicas, also with the theoretical recombinants of V plus P, P plus W, and V plus W. In a way, you can select more than one and plot all on the same graph. So this was one of the suggestions. Yes, I will likely add a graph to show IC behavior. I've written the behavior, like as something goes to infinity, this goes to 0, etc. But a graph really helps. Just to show you how it works, I can select a few. Let us select NIV and NDVW, the most interesting one. Maybe this, and compare for CCL4. So I run here, and you can see that V plus P, which is the green curve, it fails to fully suppress innate immunity. While if you have only the W protein without V and P, it's enough to shut it down completely. And when you come down with the exercise, there are a few guides on, for example, look at the equation. What do you think? What do you expect will happen if this? Check on the graph, do this and that, etc. In the last exercise, I discussed that here. So in the exercise 5, I say, yeah, this is already the answer because I've run through it. So ideally, the person meets this and then run, and this is the answer. The target of the drug would be the W protein. So any chemical species that binds to W protein and prevent it from binding to STAT, suppressing all sorts of pathways in the diagram, would at least not shut down completely innate immunity. Because even though you have V and P, as the graph shows, VNP failed to suppress innate immunity completely. So you can, for example, combine a drug targeting W protein with a drug that stimulates production of cytokines. I don't know what it will be, but you could, I think, getting rid of W protein would be necessary to treat Nipah virus, I think. And that's what I've done of new in my Colab notebook. So time for questions now.
It might be nice to explain what doesn't behave the way you expect qualitatively. In other words, I would have thought if I turn this off, this happens, just to give an example of that.
Oh. Let me think what was I think, yeah, so let's take interferon production and secretion, Vmax2. So what I've done to check was I select interferon beta mRNA and plot, let's say, just NDV. So what I expect is if I increase or decrease the transcription rate of interferon beta, I should see, because of all the feedback loops back into the mRNA, I should see an appreciable effect of the transcription rate of interferon beta on its mRNA. And if I change here, you see that there's like no change at all. I mean, I'm changing multiple orders of magnitude here, and I see no appreciable effect on the levels of interferon beta mRNA. Because it doesn't care. In the end, it doesn't care, because in the equation, as the parameters are set, the way the parameters are set, the basal rate of production of interferon beta mRNA and the decay, they are way, way, way more important than IRF7. So this is something that kept me wondering if they actually used the mixed units parameter instead of the converted units. Or I don't know, or they didn't notice it, or they didn't bother, I don't know. But this is the kind of unexpected behavior I told. I mean, if you increase the production,
Vmax2 is where in the circuit?
Vmax2 is this arrow here.
The production of interferon beta, extracellular interferon beta.
Yeah.
So if you plot the amount of extracellular interferon beta and you change Vmax2, does it change?
Yeah, I'm pretty sure it does. Let me show you. Let me reset this more or less. 10 to the minus 15, which is worrying in my opinion. The values still don't make sense. As I increase here, 10 to the 8, like multiples order of magnitude, you see it was 10 to the minus 15, now it's 10 to the minus 12.
So it does change.
Does. Okay. Yeah. It's a worryingly low value of interferon beta, because 10 to the minus 12, there's no parameter you can multiply that in the circuit that is capable of triggering anything.
So it's basically zero. So effectively, the interferon beta does nothing.
That's what I think. but I checked the parameters over and over and nothing. And the converting the units, I expected to help that because I am most of the parameters I'm multiplying by 3,600.
And they don't plot their time series, so you can't compare to their results.
They don't plot the time series with the raw values. They normalize by the maximum. So I have no way to know if they are getting 10 to the minus 15 like I do.
And they don't give their raw code. Their code was written in MATLAB, or
Uh, I think I can check that if
Well-
not now later. But
we don't have to do it right now, but if it's,
okay.
you might look, because something is strange. I mean, seems that you've been very careful, but I don't know what to tell you. It's hard.
Yeah, I understand. I understand. I was very depressed yesterday doing the presentation and thinking about that maybe I failed to replicate, and there's a high chance that I failed to replicate completely. So
Well, I wouldn't say it happens every year, but we've had many years when people have found mistakes in the papers they tried to replicate. I would say every other year, somebody finds a major error in the paper that they replicate. So it's not unusual to discover that the paper has a big mistake. I'm not saying whether it does or not in this case, but it's not unusual to have the paper have a mistake, major mistake.
yeah, okay.
This is why reproduction is important.
Yes, and people don't give the value it needs.
And it's a good reason why you should always give your raw data as well as the code you ran, so that people can check. Very
Yeah,
good.
it's true.
Any other questions for Pedro? I know we were running a little behind, and I don't want to have people run out of time. Okay, thank you very much, Pedro. So next we have Ibrahim speaking, I think.
Okay. I'll try and keep it a little bit shorter just because we've been it's been, I guess, a lot of the presentations before have been pretty long, and I don't really want to bore you guys with a lot of the same information as last time. So just a reminder, my presentation is modeling predator-prey interactions with the Lotka-Volterra model, and additionally adding other more realistic parameters. My name is Ibrahim, and let's move on. So this is just the overview. This is the paper that I've been working on. It's called Predator-Prey Models Review Some of the Recent Advancements. And there's my Colab link. We'll go over that at the end, hopefully. So some biological background is that basically in biological systems where predators and prey both coexist, the interactions between both predators and prey dictate how the population densities progress over time and what their steady states will be. So it is important basically if we want to analyze real life ecosystems, to be able to create a mathematical network model to help us better understand how different characteristics and features of the ecosystem can impact the predator and prey populations over time, and also just in the extremes as in the steady state. So back in a long time ago, I want to say the early 1900s, Lotka-Volterra basically designed the simplest mathematical network model for predator-prey interactions. And here basically we'll see the ordinary differential equations describing the two. X is the prey, and then Y is the predator. And basically the I accidentally wrote the wrong coefficients, but I'll just replace those with what they're supposed to be. A here is actually the intrinsic growth rate of the prey population in the absence of predators. So basically it represents the rate at which the prey population increases under optimal conditions. This is just the natural growth rate of the prey species. And then B, this is also wrong, my bad. B is the rate at which prey are captured and consumed by predators. And basically it is symbolic of the effectiveness of predation of predators on prey species. So what's important to note is that the growth of the prey species is just dependent on the concentration of the prey species. However, the decay of the prey species is dependent on both the predator concentration and the prey concentration, because the prey species, because if there's a low amount of prey species, there will be lower predation because the predators find it more challenging to find the prey. That's what this is meant to simulate. And that also simulates a lower effectiveness of being able to catch prey. So here, the predators population is quite similar. In this case, C is the death rate of the predators in the absence of prey. This is just the intrinsic decay rate. And then B is the effectiveness, which is D here, which is the effectiveness of predation on prey. It's similar to B. However, here we can see that the decay is just dependent on the population of the predators, while the growth is dependent on the population of the prey and the predators. This is for a similar reason. It's because this also incorporates the intrinsic, I guess, the reproductive rate of the predators. That's what this is also meant to simulate, which is why it is also dependent on the population of the predators. So it's almost like if they can eat and they can reproduce, then they will grow. So that's just the simple Lotka-Volterra model. And here we've kind of been over this in class, so I don't want to go too deep into it. But basically, we kind of see this oscillatory system where predators and prey can both coexist, and their population rises and falls, which makes sense because if there's a high prey population, the predator eating effectiveness will increase, which will cause the prey population to decrease because they keep getting eaten. And when there's low prey, then the predators will start dying because they're not able to capture food and eat as effectively. And then we get to this scenario where there's low predator and low prey. And if there's low predator and low prey, prey will be less likely to be predated on, and they will grow, and you get this kind of cycle. So while that is valuable, it is even more important to to, I guess, build upon that, because it's kind of a basic simulation. So one of the important additions you can add to the basic Lotka-Volterra model is what's called the alley effect. And the alley effect is basically a biological phenomenon that describes the correlation between prey population size / density and the growth rate. And basically what this means is it occurs when the population of the prey species is below a certain threshold, therefore making it difficult for them to reproduce and survive. Which basically means that at the prey species below a certain population threshold, there will be a different rate or a slightly modified rate. That can either be lower but still positive, and that's called a weak alley effect. Or it can be lower and negative, and that's called a strong alley effect, which means that eventually, if the prey population goes below a certain threshold, the species will have a negative growth rate, causing the species to die off. Next, okay. So just some of the mathematics for this though. So simply the alley effect is basically represented by modifying the growth function of the Lotka-Volterra model, which is, we recall, which is basically this portion. This is the growth of the prey. And you can do that, you do that with this type of equation, which is dependent on the intrinsic growth rate and the carrying capacity of the system, and also this m. And this m is the alley effect coefficient. And if m is positive, you have a strong alley effect if m is positive. You have a weak alley effect if m is negative. And that's because if m is negative, this function will always be positive. So even if it's technically a lower reproduction rate, it'll still be positive. But if m is negative, this could be a negative coefficient in certain scenarios, causing the growth rate of the system to be, of the prey to be negative, which would cause the extinction of the species. So basically, we are basically just adding this into our system. These are the letters just have changed because, I don't know, every single person who does Lotka-Volterra model has different letters for everything. But basically, this is still the predation rate. This is the predation effectiveness for prey. This is the predation effectiveness for predators. This is the intrinsic decay rate of the predators, and then this is the modified growth rate, which, as we said, is still dependent on the intrinsic growth rate of the prey, but also now it's dependent on this alley effect coefficient. So if you do the mathematical analysis on this, you can actually discover 3 fixed points. The first fixed point and the second fixed point are 0, 0, and 1, 0. Well, when we take a look at the graph, we can actually see this. But these are unstable equilibria, as in they're not attractors, repellers. I think they're repellers. And then you can also calculate the stable fixed point, which is dependent on the intrinsic growth rate. Well, so actually you can do this. So when you actually do the calculations, there are stable fixed points in all 4 quadrants, but because this is a real life scenario, the only valuable fixed point is the one in the first quadrant. So the stable fixed point is dependent on the intrinsic growth rate of the system, as well as the A, which again is also the, I believe this one's the decay rate of the predators. Sorry, A is related to the alley effect in this scenario. If we take a look at the graph with the weak alley effect, so with the weak alley effect, as in the M is negative, we can see, I guess, what the alley effect is trying to describe. And the alley effect, like I said, is describing that the prey growth rate is lower than normal at a certain, below a certain threshold. So here we can see the growth rate of the prey is pretty high, but once we go below a certain threshold, which is here, the growth rate of the prey is lower. And that's basically what we're seeing here. And here we plot some of the fixed points. So there's one here at 0, 0, and there's also one here. This is the calculated fixed point. So just a disclaimer, the way that I'm displaying these fixed points is not completely robust. It's kind of an estimation. I'm basically just looking through both the derivative of x and the derivative of y, seeing when they are both below a very low number. And if they are both below a very low number, it will plot that as a fixed point. So you can modify that value, like the threshold value, and you get, yes, more detailed approximations of the fixed point. So right now I think the threshold is like 10 to the minus 3. But if you make it 10 to the minus 4, you'll just get this as a fixed point. But if you make it like 0.5 times 10 to the minus 3, you'll get a fixed point here, a fixed point here, and a fixed point here. So I kind of added this into the code just because I wanted to make it dynamic so that if you change the parameters, you could still get fixed points, but you kind of have to play with your threshold to make sure you get the expected fixed points. Because also you could make it like 10 to the minus 1, and if it's 10 to the minus 1, you'll get like a bunch of fixed points along the null line almost. So here we're basically looking at the weak alley effect, where below a certain threshold, which in this case is very low, you get a lower growth rate for the prey population. So a strong alley effect, on the other hand, what we'll see is that, and just a reminder, the strong alley effect is supposed to give us a scenario where below a certain threshold, the growth rate is negative, and that's exactly what we see here. Basically, if the prey population is above this threshold, it's positive. But if it's below this threshold, it's very tough to see because of the bounds in the specific graph. But if it's like here, 0.001 or something like that, the growth rate will be negative, causing the whole system to spiral into extinction. Not necessarily spiral, just go straight to extinction. So that's the alley effect. So the next one that we wanted to look at was the fear effect. And the fear effect is a way to simulate how prey feel fear or experience fear towards predators. And therefore, if they experience fear, they will actively try and avoid them. And this idea kind of has some consequences associated with it, basically meaning if the prey are scared of predators, over time they will be less likely to go out and reproduce because they experience fear with the predator, and therefore their growth rate will be reduced. So here we can see that in the mathematical model. So here in the growth section of the prey, we see that now it's dependent on the intrinsic growth rate as well as this fear function. It's called fear of SY. And basically, the fear function is any function that decays to 0, basically, as time progresses, trying to simulate the biological reality that as the predator population increases, you get more scared prey individuals, which will ultimately lower their growth rate. So here, basically, S is the fear parameter. It kind of is an explanation of how scared they are. If S is greater, the prey are more scared. If S is smaller, the prey are less scared. And then, so this is added to the positive function for the growth rate. DNA are just simplified versions of the alley effect coefficients. They just simplified, like, this is, I think A is like M over K times something, and then D is something similar of the sort. So it's just, I guess they didn't want to write it out, so they just simplified the equations. G of X in our scenario here, we're just going to be substituting for with, so G of X could technically be any, I guess, relationship that you want the predator, the preys to have with the predators. But in our case, we're just going to be following our simple Lotka-Volterra G of X, which is by. Yes, sorry, bx. No, sorry, yeah, bx. And then here, the y is basically the same. Negative n is the intrinsic decay rate of the predators, aka the death rate. C is the predation effectiveness, and then gx once again is just going to be the same here, which is just x. So here, if we take, I guess as we would expect, understanding what the fear does to the system, we can do a little parameter scan, changing. I screenshotted this. I guess I screenshotted a very, very low resolution. But basically, this green is a higher S, and then purple is a lower S, and then blue is the lowest S. So basically, what we can see if we do the time series parameter scan, we basically will see what we kind of expect is that as time increases, with with a higher fear, the prey get more scared over time, and therefore have a lower growth rate because the predator population is increasing over time. So the higher the fear, the less likely the prey are, I guess, to go out and reproduce, and therefore, when there's more predators, therefore causing the prey population to kind of dwindle, which is kind of interesting. So here we can do a little phase plot of a scenario with low fear. And if there's low fear, I guess in this specific scenario, we get the prey population going to 0 and the predator population stabilizing. And this is because in our scenario, in our equation, we're also simulating such that the predator is not dependent solely on eating this specific prey. It can, in fact, eat other prey. So even though the prey population dwindles to 0, the predator population doesn't necessarily go to 0. But here we see if there's higher fear when S is equal to 3 in comparison to S. Sorry, I should have run at the top, but this is S equals, I think, 0.2. When S is equal to 3, the prey are very scared of the predator population, and the predator and prey population both approach 0 as an extinction. So actually, let's do the demo right now before I go over just the conclusion and stuff. Fortunate to rerun everything. Okay, but while this is running, I'll also just try and go over some of the new stuff that was added, just to save some time. But basically, the first thing that was added is I created a little scenario where you can play around with just the basic Lotka-Volterra model by modifying A, B, C, and D. And if you modify,
you can't see it if you're.
Oh, you can't see it?
No, I see a PowerPoint.
Oh, my bad. Can we see now? Okay, sorry, my bad. Fortunately, I don't think I did too much. But yeah, basically, here we're just taking a look at the Lotka-Volterra parameters. You can change A, B, C, and D, which are described as were in the PowerPoint, as the intrinsic growth rate, the predation efficiency, the intrinsic decay rate, and then also predation efficiency here. If you modify it, just like kind of how we did in class, I don't really want to go too deep into this. But for example, if the growth rate of the predators is higher, if the growth rate increases, we should expect to see a shift to the right. I guess it goes up also because if there's higher prey concentration, you also get more predation, or more effective predation. So the predator population also goes up. So this is kind of what we did in class. I don't want to go too deep into it. Then for the alley effect, it's a little bit more interesting because we also can plot the time series to see if it matches with our expectation for the phase plots. So I have some exercises here that we can take a look at. This is the weak alley effect exercise. Plot it again. This is perform a plot. Yeah, so here we can take a look at the plot. We don't need, we can modify our bounds. Since it seems like nothing's interesting beyond maybe 2, we can do our, oops, this should be 3. Our X bound can be 2. And then it's also printing the expected fixed points here, which is here and here. Also, so I don't have a, you can't necessarily modify the sensitivity of the fixed point finder in this. Maybe I'll add that actually before I do the final submission. But basically, if you modify, so right now our sensitivity is only picking up 2 fixed points, which is at 0, 0, and the calculated one, which is what we expect. But we also, if you remember from the slides, we expect a unstable fixed point at 0, 1. If I change the sensitivity to, let's try 0.7 times 10 to the minus 2. See, we are actually generating more fixed points. So it seems like before we even get this fixed point, these points close to the calculated fixed point are also showing the best fixed points. But if we change it, just try 1. So this is, I guess, a limitation of the approximation strategy, is that your actual fixed points could possibly be detected later than points around other fixed points. So here we can actually see that we are kind of getting a cluster of fixed points around 0, 1, which is what we expect to see. And then also we're getting a cluster around this calculated value. Which I think is, I guess, it's valuable to understand. I guess you should just assume that points clustered together represent a single fixed point in this. So we can also take a look at the time series plots. The time series plots are, I guess, interesting because we can see if it matches up with our expectation from the phase plots. So for example, let's pick 1, 1. We start here 1, 1. What should we expect? The prey population, we should expect it to decrease and then overcorrect and increase slightly. And the predator population, we start at 1, 1. We should expect it to decay eventually to the stable fixed point. Let's see if that's what we get. Here you can just modify the time also, because sometimes the time is affects where you want to. Yeah, so here, this is what we thought. The prey decreases, overcorrects, and then approaches equal the steady state. And the predator population is what we expect here. I guess, yeah, a slight increase, which I guess follows this line over here. A slight increase, and then it drops down and approaches the steady state. Now, more interestingly, we should check to see if the alley effect is actually working. So we should try a low prey population and see if the growth rate is different at a lower value. Because here we can see the growth rate is constant almost. So if we choose prey start at 0.001, let's try this. So we should expect to see a low growth rate for a very short amount of time, followed by the rapid growth rate again, which is what we see here. So we see low growth rate followed by the rapid growth rate, and then it overcorrects. So here we're glad the alley effect seems to be working as expected. We're happy. So next is the fear effect. Yeah, so the fear effect, it basically functions similarly. We have an example that I made, one specific exercise. Let's see if we can do that one. It should set it to my previous one. Yep, okay. Yeah, so here we are looking at a scenario with a lowish fear, and the lowish fear is causing prey population and predator population to have a stable fixed point here, and then also an unstable fixed point here and an unstable fixed point here. So this is an example of a stable circuit or a stable phase plot for both the this is a weak alley effect, I believe. No, sorry. Yeah, sorry, for a high S. My bad, I was thinking about the wrong thing. This is for high S. And then also we have time plot as well for this, where you can verify that what you expect to see from the phase plot. I think it's valuable in that way. But other than that, that's basically it for this. Let me switch back to my PowerPoint. Okay, so some of you may be wondering, where is cannibalism? Here. So this was an issue that I was running into the paper. So the paper had two more additions to the Lotka-Volterra model. One of them was cannibalism, one of them was immigration. The problem with these two systems is that they didn't add on to the previous ones. They kind of just had them like laying there. They just said, also, by the way, cannibalism is cool. You can have this C of X or C of Y function, and everything works out fine. So that worked out nice and fine. I've created it on its own. The problem is when I tried to add the cannibalism function to the existing model, I'm sure it technically works, but I was not able to find any parameters that could give me anything meaningful. I mean, maybe before Friday, I'll just keep working on cannibalism specifically because that one was easier to implement. Just try and get something working. Any possible, I guess, parameters, that would be, I guess, better than nothing. But that's basically, I guess, the issue I was running into more recently. I spent a long time trying to get cannibalism parameters to work, doing parameter scans all over the place, and still nothing was working out well. But yeah, other than that, that's basically everything for the project, the Google Colab note, and then the reference as well. That's basically it. Any questions?
So who's cannibalistic, the predators or the prey?
So in the paper they do you can technically do both. They say you can do both for both predators and prey, but the paper itself has a cannibalism function for predators specifically, and also more annoyingly is that in the paper they don't have parameters for it. It's kind of just an exploration of whether or not you can do it. They don't actually do any simulations or graphs with it. They just say you can add this C of Y function or this negative C of Y function in the predator rate equation. They don't.
Do the predators always eat each other, or they only eat each other when there are not enough prey?
So in that one, they will always eat each other. But theoretically you could change C. Well, they give a couple examples of different cannibalism functions. I think one of them is always eat itself, and then another is, I think, what you mentioned, which is will eat themselves in certain scenarios.
So basically, it's an LA effect on the predator instead of on the prey.
Yeah, that's what it does tend to be. Yeah. Like I said, I'll keep playing around with the parameters, see if I can get anything that works, I guess.
People might do the same thing. Meet up with the person you met up with last week and go through another time together. So, William, you're at the end today. Thank you for being patient.
I worked on the P53 and DNA damage, and I'm going to go into it now. My name is William Cesaretti. I'm a computer science major. Yeah. Oh, okay. So here's a little overview of what I'm going to be talking about. I'm going to go over I'm going to be going over the biological background, and then like the motivation and some goals I had for the notebook, the mathematical background of the paper, some key results that I found, issues I faced, and future work that I potentially want to implement. My Colab notebooks is here. I'm going to go over it after I go over the slides, and then some key references. These are just 3 different papers that I cite. The main paper that this notebook is modeled after is the first one, exploring mechanisms of blah, blah, blah. I'm not going to read that, but no. Okay. So here's a quick biological background slide because I think I had a lot last time and I just wanted to make it short. So DNA in our cells is under constant stress from many environmental factors, and so our cells have created complex mechanisms to combat the this DNA damage. And one of the mechanisms is this the protein P53, and P53, I guess, sort of lives in or works with MDM2 to repair DNA damage. So MDM2 and P53 are both proteins. And as you can see in the picture on the left, MDM2 and P53 will go inside the nucleus and into the cytoplasm, and vice versa. And researchers have found levels of P53 and MDM2 rise and fall in pulses, and I wanted to investigate these pulses, and that brings me to the motivation and goals. So P53 is in the system of P53, MDM2, and DNA damage is important. So I wanted to create an interactive walkthrough of the system, try to make it like not boring, and I wanted to maybe draw conclusions that the paper has not did not make clear. So I wanted to show very clearly DNA damage's effect on pulses. So I'm going to get into the math. So I have provided, or the paper provided, this schematic diagram, which was very helpful to me. and to people reviewing my project. So I guess I can go over, maybe I'll go over the schematic, or maybe if people can just look at it. Essentially, the solid arrows mean that concentrations are flowing, or it's sort of where concentrations are going. So for example, from a source to P53, this is what this arrow means. Or from MDM2 in the cytoplasm, MDM2 goes to MDM2 in the nucleus via this arrow. The dotted arrows mean that it's activation. So MDM2 in the cytoplasm with this arrow activates the production of P53. Okay, so and notice how DNA damage only activates one thing: the degradation of MDM2 in the nucleus. So this is the way that the researchers chose to model the biological system. And here are the three main ODEs, or three only ODEs that constitute the three main substances besides DNA damage. So maybe I can explain, I'll just explain one of them really quick. So let's just take P53, for example. So the first ODE, dp53 over dt, is the essentially the rate of P53 is the basal rate, which is K prime S53. So the basal rate is the rate at which P53 is being produced in a healthy cell with no DNA damage. And then this is the Hill function. So this tells you, okay, if MDM2 is high in the cytoplasm, then P53, then this term will be high. So, but if MDM2 is low, then this term will basically be not important, and it will be low. And also notice here how K double prime is the S53 is the maximum value of that this term can take, since the Hill function, the maximum value of this Hill function is 1, because if MDM2 is cytoplasm, if the numerator is super high, that means that the denominator is also super high, so it'll be infinity over infinity, kind of, and it would equal 1. And lastly, this is the degradation term. So for P53, we can map this to here, to this arrow. And KD53 is the decay constant. Well, it's not actually a constant, because this D53 depends upon this dotted arrow here, MDM in the nucleus. So we calculate this D53 later. And then similarly, the second equation is very similar, but maybe I can note one thing here. So MDM2 in the cytoplasm and MDM2 in the nucleus, it's not transpose, it's transport. Okay, the concentrate, so MDM2 in the cytoplasm and MDM2 in the nucleus transport to each other their concentrations. So we can see this with these terms. So MDM2 in the cytoplasm has a decay term of Ki times MDM2 in the cytoplasm. And this is not a decay term, this is a transportation term of MDM2 in the cytoplasm to MDM2 in the nucleus. So this is representing this arrow here. So, and you can see the exact same term here, Ki times MDM2 in the cytoplasm, but it's positive. So the signs are flipped because it is taking in from the cytoplasm. I hope that's clear. And vice versa for this term and this term. So this is the other way around. I think those are all the main things I would like to say about this. So moving on. Here's a little bit more mathematical background, because the previous three ODEs just described these three proteins, or two proteins, and two of them are in different compartments. But DNA damage affects the decay of MDM2 in the nucleus. So if we go back to MDM2 in the nucleus, that is this term KD2 times MDM2 in the nucleus. So right here, KD2 equals K prime D2 times 1 plus DNA damage. So they are sort of representing DNA damage using the Goldbeater-Koshland function, a heap side step function, which is just a binary function. And this is the change in DNA damage is just using the heap side function with the repair rate, because over time the DNA will be repaired. And I think that's all I want to say. KD53 is the decay constant of P53. And this is important because remember, this is here, and this is affected by MDM2 in the nucleus. So this is how they model it. I honestly don't fully understand this part, the Goldbeater-Koshland function. I just know that they use it to model oscillatory systems. So here's a little goal. So my goal was, well, sort of the main goal was to make a notebook that is educational, like I previously said. But I think that we figured out that we wanted to figure out the conditions at which pulses occur and at which they stop and why. So here is just the raw. This is just a raw time series of the initial conditions proposed by the authors, and as we can see, when DNA damage here is introduced at time equals 50, and when it's introduced, I stole this off of the previous slide, so that I made, it's not labeled. All the other graphs are, but at time equals 50, DNA damage is introduced. This causes MDM2 in the nucleus to decrease right here. So DNA damage comes in, MDM2 starts to decay more. This causes a rise in P53 and MDM2 in the cytoplasm, and then they end up MDM2 in the cytoplasm then transports its concentration to MDM2 in the nucleus over time. This causes MDM2 in the nucleus to rise, P53 to drop, etc. So that is how a pulse is created. But here are some of the graphs and the experiments that we ran, or I ran. We changed when I say we, it was James helped, and Pedro, thank you. But we changed the amount of DNA damage to be constant. So initially in the paper, DNA damage comes in at time equals 50 and starts linear decay. So in order to and that's an extra variable that we did not want to handle. You sort of just want to we wanted to make something a constant, so we could further explore the model and understand it. So we set DNA damage to be constant, and this allowed us to, or this allowed me to change the level of DNA damage, or this allowed the user to actually change the level of DNA damage and plot the time series. So when DNA damage is 1.4, we get many little oscillations. And look at our phase portrait in the bottom left with DNA damage at 1.4. So this is plotting P53 and MDM2 in the cytoplasm. And we are not getting any loops here. So each black dot is, you remember phase portraits, I think we all do. I just made it more clear to the user. Each black dot represents a set of initial conditions of P53 and MDM2. So if you take one of these lines, trace the color all the way, it will stop here, which is the steady state value, right? Which is like, if we keep running these lines forever, it's going to be around here for each of them. But if we increase it to 1.5 from 1.4, we instantly see these huge pulses, repetitive over and over and over. And this tells, and also in our phase portrait, we see loops. So unlike here, we saw no loops. Here, you can sort of see, so it's kind of hard. I was trying to make it easier, but there's this one clear loop. Brown is a clear loop going around and around, and a loop in a phase portrait indicates oscillations. So obviously there's something going on between 1.4 and 1.5 with the DNA damage level to make these oscillations happen. So to zoom in even further, we plotted the bifurcation. We plotted a bifurcation plot of DNA damage, zooming in. So we zoomed in the x-axis a lot, and so this is our y-axis is p53. So we were plotting p53, the steady state of p53, and changing DNA damage here. So when DNA damage hits around 1.4 and change, like 1.41 ish, then we sort of see the split into two, and this is called a HOP bifurcation, or HOP bifurcation, or HOP bifurcation point, I think. And this represents oscillatory responses. So we were just plotted the maximum value of p53 and the minimum value of p53, and yeah, so this graph shows that around 1.41, the constant steady state that we used to have with p53 changed to an oscillatory response. And so now I wanted to take this information that we learned from keeping the DNA damage constant and move it to DNA damage that is decreasing. So I allow the user to change DNA damage. So this is DNA damage at 3.9, DNA damage at 2.1. So for this first, or maybe for the second graph here, DNA damage is relatively lower than the above graph. So when DNA damage starts and it's decaying, we only get one pulse or one oscillation. But when we increase DNA damage and I made it so the y-axes dynamically change with the amount of DNA damage. When we increase the amount of DNA damage, we will see two pulses, okay, because it takes longer for the level of DNA damage to hit this 1.4 spot, right? It's DNA damage is decreasing, and then once it hits this 1.4, then everything comes back to steady state. Hopefully that is clear. So those were sort of my main takeaways. I have a bunch more plots, but honestly, I didn't really want to talk about them in the PowerPoint. So some of the issues I faced were changing some of the parameters, like computationally was oddly hard sometimes because of the way the model was coded. But I don't think I faced as big of problems as others. And I would say understanding the mathematics behind the way they plotted DNA damage was a little bit confusing, and the way they modeled it and how it affected the system was confusing. But yeah, so my conclusion was that pulses are related to the DNA damage level, and ended up figuring out exactly where what level of DNA damage created these pulses, and now a user is able to understand this interaction throughout Colab notebook. There are additional phase plots, etc. Different parameter changes in there as well. Some future work. I have to do the DOI publishing. Oh, this didn't update. Examine other models in the paper, change the mathematics of the model maybe, and also add exercises, I think. I mean, maybe I'll just open the Colab now. Yeah, so I'll open this. Okay. Okay, I just see a lot of comments in the chat. It's making me nervous. All right. Okay. So I think this is the one I just opened. Yes. Okay. So you open it up and sort of a biological background. I'll just go over the stuff that I changed and what I think is important. Elmer added the Wikipedia page for you. Mathematical model. This is all sort of the same. Added the schematic diagram, which I think is very helpful. Okay. So, I have to install this. So I have these, I think everyone has this where you can drop it down. So once this installs, so essentially the big change was, thanks to James, was modeling with constant DNA damage. So the initial model had that linear decay of DNA damage, but this just added another variable that we had to work with. So it made more sense to just so essentially I just run the model and plots the constant DNA damage, and then it shows two pulses. Since this is the first time the user is seeing these pulses, I wanted to just make it simple and explain to them what they were and whatnot. Going down, this sort of took a while to figure out how to code without the duplications and of the graphs and stuff. But so this is the user can sort of graph now. So 1.6 you can see, and then you can keep increasing to 3, and everything updates, and you can see the loops, and I think it's pretty cool. If you make it super small, I mean, nothing, you could, this makes sense. So then I plotted the bifurcation diagram. So this was just plotting the steady state value of P53, and then it was actually plotting the minimum and maximum values of P53, because after a while they change, they change. And then I get into decaying DNA damage. So this is just what I had before. Now the user is able to change the amount of damage here and see. Here I zoom out even more, allow the user to see just a longer time frame. Now I look into changing the production rate. This I didn't go over in the slides. So this is changing the basal rate of transcription of P53. And it shifts the graph to the left, which is what we expect. Here's another changing one of the scalars of P53, or scalars of one of the scalars of the terms that contribute to the change in P53. This again makes the graph more condensed, which makes sense, and because P53 is being created at a higher rate when the scalar is high, and so all the reactions all the math happens faster, I guess. I don't know how to explain it. So then I looked at phase plots, and this allowed the user to see the steady states of each, or of different combinations of variables. And what was interesting here, or not really interesting, what was annoying was that you have to think about each of these phase plots as MDM2 as the variable that is not being plotted is fixed and it's sort of frozen, which was annoying, but I don't think there's another way to do it. I mean, I think the only way we thought is it had to be 3D, which I actually did see online that you could do, but maybe I can try that. So then here's another one. And you can see that some of the arrows are leading outwards. So it's kind of weird. Let's see what's going on here. And that's because there's two stable points or two fixed points. But this one is not actually here. It's due to the freezing of the MDM2 in the nucleus. And yeah, and here's the last one, MDM2 in the cytoplasm. And I mentioned the nucleus, since they're transporting to each other, it's a pretty direct, it's a direct relationship. So there's just one fixed point. Here's my conclusion, references. Yeah. Thank you.
Great. Comments or questions for William? The one thing I was thinking of, I put a few things in the chat, is that it might be worth explaining what the role of P53 is biologically.
Okay.
That P53 is a master regulator of cell death. And so essentially, if you get too many spikes of P53, it's telling the cell, you're damaged so much, you better die. Because if a cell is very badly damaged, you would rather have it die than recover, because that's how you get one of the main ways you get cancer is when cells don't die when they should from DNA damage.
Okay, yeah, that I definitely need to add that if I don't have that. Yeah, thank you.
So I think I don't know how many P53 spikes it takes for the cell to decide to die, but it's an interesting. It doesn't affect the mathematics of what you're doing, but maybe for motivating the story a little bit, it's important. I do think that the way you display things works very well. Ibrahim, go ahead.
I can't hear you. Hello?
Ibrahim, go ahead. He put in the chat, he says, try using quiver plots, and is that different from stream plots? You mean to try to do 3D plotting of the flow? That's possible, I think. I don't know.
I saw on Tellurium that you can. Or was that not the right?
It may well be. I know people have done it. I know people have
pull it
done
up.
3D plots in the past for flow. I didn't ask people to do it because it's a little more complicated, and I don't remember the package to do it with. But if you can get it to work, that's very interesting. Okay. Any other questions? or comments? Well, I think people are tired after all of that. I really do appreciate everybody taking the time to work on this together. I think everyone dug into these projects a lot. It's a lot to do in a semester, especially when you're doing it on your own. I'll copy the chat comments and make them available to people. And I also may try to, if people have comments for each other that you can send around the way we did before, maybe that'll help people clean up a few last things before you do final submissions. And again, I want to thank everybody for your patience and willingness to explore the semester. Somebody says, is it still possible to submit a homework assignment? If we get the homework assignments in by Thursday, I will try to grade them. But I'm going to be at a conference in the Netherlands, so I'm not going to have a lot of time to do grading. So it's going to be, I need to get them in as soon as possible. I'll do my best. I know it's a lot for people. I would concentrate on making your projects, especially if you have a DOI, I would concentrate on making your projects as useful and inciting as possible so that when people see them, they want to run them. You've got lots of great ideas here. And I think it's nice for you if people could look at those projects and appreciate the effort you put into. Any last comments or questions before we break? Okay, well, I want to thank everybody again for sticking it out, and I hope I look forward to seeing the final versions of the projects and seeing you. I assume I'll see many of you going forward in various ways. So thank you again. If I don't speak to you before the holiday, have a great holiday.